Hello aspirants, welcome to Sri Ramayas. I am pleased to announce that we are starting a crash course for prelims 2023. So the target in this crash course is that in 30-35 days we will cover the major portion of the syllabus so that it is beneficial for you for this prelims. So in prelims, there is a general tendency that in prelims we think that it is mainly current affairs based, but if you look at the pattern of which UPSC is asking question. So you will realize that yes, the question might look like current affairs based, but the question requires your understanding of the static portion quite well. So I'll just show you through an example, like in biology and biotechnology, there are a lot of news with respect to gene editing. So you would have heard about CRISPR-Cas9. So this CRISPR-Cas9 is being used in many ways to bring gene therapies. But what is this CRISPR-Cas9? UPSC will ask you just a basic question, what is Cas9? Do you understand what is a restriction enzyme? Do you understand what are the different gene editing tools? So you need to have a basic understanding how do we do gene editing. But before that, you also need to understand what is a gene, what is a DNA. So let us just try to understand this DNA and gene and how do we do this gene editing. So if you look at the structure of DNA, so the DNA structure is very famously known as the double helical structure. So if you look at the structure of DNA, it has this helical structure. And in this helical structure, you will see that these are known as the strands. So this is one strand, this is first strand, and this is known as second strand. So you would have seen those spiral staircases. Our DNA just looks very similar to those spiral staircases. So these strands are just like the railings, and these strands are built by alternate layers of sugars and phosphate. They are built by alternate layers of sugar and phosphate. And what do you see that in these strands there are some bases, there are some nitrogen bases. What is the significance of these nitrogen bases? Just as computers, they understand the language of zeros and ones. Computer understands the language of zeros and ones. So our living organisms, they have a different coding mechanism. They use what are known as ATCGs. These are also known as the nitrogen bases. So how they are present in this helical structure? So they are present in this manner. So if you have A on this side, then let us say you have T, C, G on this side, then correspondingly we have a rule that on the other strand, that along with A only T can come, and along with C only G can come. So if there is A on this side, on the other side of the strand there will be this T nitrogen base pair. If there is T on this side, there will be A on this side. If there is C on this side, there will be G on this side, and if there is G on this side, there will be C on this side. So in this way our whole DNA is constructed. So this one group, or the A and T that you are seeing, is called as base pair because it has two sets. So that is why it is called as base pair. So in news you would have heard that we are using CRISPR-Cas9 to do base editing. So what is this base editing? This base editing means we are trying to change these bases. So in any gene therapy that we talk about, what we are trying to do is essentially change these things or manipulate these nitrogen bases. So whether you hear about CRISPR-Cas9 being used to treat Alicia. Alicia is a girl in UK. So it is a very famous case that Alicia is a girl in UK and she is suffering from an autoimmune disease. So what is happening in her body? Alicia is suffering from an autoimmune disease. So what is happening in her body? That you would know that in our body we have the white blood cells. white blood cells are our defense mechanism. So if any antigen or any pathogen attacks us, like a virus or a bacteria or like COVID, if they attack us, our antibodies get activated, our WBCs get activated and they start making antibodies. And one of the type of WBCs is your T cells. These T cells are known as killer cells. These T cells kill the pathogens. So these are your defense system's killer cells. So what is happening in her is that her T cells are killing her body's healthy cells. So her T cells has somehow got mutated and her T cells are killing her body's healthy cells. So what is done in her case, that using CRISPR-Cas9, what we are doing? We are doing base editing. So what we have done, we have extracted the T cells from her body. And in lab, we have gone for base editing. Of T cells. And after base editing of the T cells are now called as the healthy T cells. And what is done now is that these healthy T cells are transferred back to her body. So when it is transferred back to her body, what we are seeing that her new healthy T cells is able to kill the unhealthy T cells. So in this way, what we are seeing that her autoimmune disease is receding. That is the biggest advantage of having some gene therapies to cure some of the diseases like this. But in lab, when we have to go for gene editing, we need many tools. What are the different tools that we need for gene editing? So now we have understood that what is base and what is base editing, but how do we do that base editing? In order to do that base editing, we require few tools. What are those tools? So the tools for gene editing. The tools for gene editing is just like our normal stationary mechanisms. So just as we need a scissor. So let us say if you want to cut or make editing in this gene, or you want to cut this particular gene. So if you have found this particular gene to be defective and you want to cut this part, and you will cut this part and you want to remove this part, you want to join these parts. So what do we need? We need something called as scissors. But in biology, we generally use what are known as enzymes. Enzymes are chemicals. These chemicals help us to do this cutting part, or these chemicals will only help us to do the joining. So in biology or in biotechnology, we use or we take the help of enzymes to do these gene editings. So there are very famous enzymes that we use. So the first famous enzyme that we use is known as restriction enzyme. This enzyme is also called as molecular scissors. Why it is called as molecular scissors? Because it helps us to cut a specific part of the DNA. So we can identify a particular part of the DNA, and when we want to edit it, we can cut those parts. But from where do we get this restriction enzyme? In nature, we see that this restriction enzymes are present in bacteria. So where do bacteria produce this? So if you take a normal case of a bacteria, so let us take this as a E. coli bacteria. E. coli bacteria is said to be a rod shaped, just for understanding, let us understand that it has some rod shape, and there are some viruses which can eat bacteria also. Those viruses are very unique viruses and they are called as bacteriophages. So these bacteriophages are unique type of virus and they attack bacteria itself. And when This is your bacteriophage. This is your bacteriophage. And inside the virus, what you will find? Either a DNA or an RNA material. And let us say this is a bacteria. This is a bacteria. So how does this virus infect a bacteria? So what it does, the virus inserts its genetic material into the body of bacteria. So virus wants to multiply, but virus is inactive outside the body of a living organism. It can get activated only when it is inside a living organism. So it attacks a bacteria and it tries to insert the genetic material into the body of bacteria. So what will the bacteria do? Bacteria are one of the first living organisms. They are the most resilient organisms. They have also developed a defense mechanism against these type of viruses. So they have developed a defense mechanism, or they have developed an enzyme. That enzyme is called as restriction enzyme. What does this enzyme do? The bacteria produces this enzyme against these type of viruses, that is known as bacteriophages or bacteria eating viruses. That this enzyme, the RE enzyme, this RE enzyme cuts the RNA at multiple places, the RNA or the DNA of the virus at multiple places. So what is happening in this case? The virus attacked the bacteria, it inserted the genetic material, but it is not able to multiply. Why? Because the bacteria has produced restriction enzymes which has cut the RNA into multiple places. So the information which this RNA or DNA had for multiplication is lost. Since the integrity of the RNA or the genetic material is lost, so the virus or the bacteria is not, so the virus inside the bacteria is not able to multiply. And this is a defense mechanism developed by bacteria. So we have learnt this from bacteria only. So initially in 1980s when we began with gene editing, so we knew about few bacteria only, or we knew about very few restriction enzymes only. But nowadays we have extracted this restriction enzymes from many bacteria, and now we have around more than 900 restriction enzymes, or the molecular scissors. So these molecular scissors we have derived from nature itself. So let us say we have got the tool for cutting DNA. Now when we cut this part, we will also need to join these parts. How will we join these parts? Again, in biology or in biotechnology, we use again enzymes to even join parts. For cutting parts you require enzyme, for joining also you require enzymes. These enzymes are called as DNA ligase. Ligase. See, this term ligase or ligation generally means what? Ligation essentially means joining. Ligation helps in joining. So where do we get this DNA ligase from? So in our normal body also what we get is this DNA ligase is required. So our body also performs some gene manipulation or gene editing. So during replication or during repair or maintenance, and during that time also our body needs what? So it needs this DNA ligase. So many organisms, they produce this DNA ligase, and we have extracted from there, and we can use it to stick two parts of DNA. So now let us say we have got the tool for cutting it, we have got the tool for even joining it, but we would also need something called as the Xerox machine for a DNA. So we would also need multiple copies of the DNA. How do we get that multiple copies? So again we use another enzyme that is called as DNA polymerase. DNA polymerase. See, almost all living organisms use this DNA polymerase. So we also have evolved from single cell, we have become from single cell multicellular. So it means that our genetic material is also multiplying. So how is that genetic material multiplying? Inside our body also we have this DNA polymerase, and we utilize this DNA polymerase to multiply the DNA. So these are the major tools by which we do gene editing. So let me take another case, that how do we manufacture insulin in the lab. So insulin, what is insulin? Insulin is a hormone which our body produces in order to reduce the blood sugar. But if any person who becomes diabetic, he or she is not able to produce insulin. So, in that case, what has to be done? Insulin has to be provided to such persons externally. So, traditionally, how were we providing insulin to people? We were extracting insulin from animals, from slaughtered animals, especially pigs. But what was the concern? First of the ethical concern was that we were extracting it from animals, from slaughtered animals. So, we were deliberately sometimes killing it also. But the other medical condition was that because we were extracting insulin from an animal source, so our body, our immune system was getting activated, or there was some allergic response to those animal sourced insulin. So, what we have done, what we do now is that we produce insulin in the lab itself. So, again insulin in the lab is produced using gene editing method, or the famous method that we use is known as recombinant method. Recombinant method. So, again this method might sound a little complicated, but this is what recombinant means. We are able to combine genes from different sources. We are taking genes from animals and we are taking genes from, let us say, a bacteria, and we can mix them and then we can get the desired result. So, what we are doing in this case is that, let us say we have taken, if you take an animal cell, so if we take our cell, so this is our cell and inside the nucleus. So, this is nucleus and there you will find the chromosomes floating. So, inside human cells, inside the nucleus you will find chromosomes. We will extract this chromosomes and we will extract the gene that is needed for insulin production. So, we know where this insulin gene is, and using again restriction enzyme we will get the human insulin gene. So, let us say this human insulin gene looks like this. This is the human insulin gene, and we want to manufacture insulin in the lab. So, what do? We take a bacteria. Inside a bacteria you will find a circular DNA, a unique type of DNA which is found mostly in bacteria. So, we will remove this circular DNA. This is also known as your plasmid. And what we are trying to do is that we will cut this plasmid also at two places so that we can fit in this human insulin gene. So, again using RE we will cut at two places, that is we are preparing this plasmid. We will cut it at two places and then what we will do, we will combine these two. So, now what we will do, we are combining this and this. So, this is what is called as the recombinant DNA. Recombinant DNA. Why it is called as recombinant DNA? What we have done, we have taken something from a bacteria and we have taken something from humans and we have combined them. So, we have combined genes from two different sources and we have got a new gene. And now what we do is that we insert back this in a bacteria again. And now when we insert this back in the bacteria again, now this bacteria is also containing human insulin gene, and that human insulin gene will start to express itself. And what will happen to the bacteria? Bacteria will eventually multiply. Bacteria will multiply in numbers. So, we will get many bacteria having the insulin gene and they will start to produce insulin, and they will start to produce insulin. And when they start to produce insulin, what we do is that we extract that insulin, we purify it and we save it in glass vessels, and that is how this insulin is prepared. So, this insulin, because it is now being produced mainly by what? The source is the human insulin gene is leading to production of the insulin. That is why the rejection rate or the allergic reactions with respect to this type of insulin is very, very less. So, this has proved to be a boon for most of the diabetic patients across the world. So, insulin production is a little more complicated. What I have done here is that we have simplified this process. See, in human insulin, you just need to know that human insulin has 3 type of chains. Human insulin has A, B, C chains. So, when the cells start to produce insulin, they produce it in what is called as immature insulin or proinsulin. In proinsulin, you will have all the 3 chains, A chain, B chain and C chain. And when insulin or in the mature insulin, what you will see that C chain is not there. So, there is only A chain and there is only B chain. There is only A chain and only B chain. So, when we are trying to produce this gene in this particular insulin in the lab, so the problem that we face that we are able to produce this, but we are not able to get this. We are able to produce all the chains, and that is a problematic, because when we are providing insulin to people, we want this one and not this one. So, how do we remove this C chain? So, the simple method that was found that we will produce A chain separately in the lab. Using this method only, we will produce the A chain separately and we will produce the B chain separately, and then we will combine them. So, we will get a mature insulin. So, this is how insulin is manufactured, and again what we are doing here, we are using tools of gene editing only. So, this gene editing tool has been used in many places now. So, you would see or you would hear that CRISPR-Cas9 is being used. So, what is this CRISPR-Cas9? CRISPR-Cas9 that you often see in news. So, CRISPR-Cas9, what is this CRISPR-Cas9? In this, the bacteria identify a particular pattern which is known as the palindrome pattern. What is a palindrome? If you look at this word, let us say a simple word like little. If you read it from this side or whether you read it from this side, the word reads the same. That is known as a palindrome. So, when a bacteria is developing that defense mechanism, so it also tries to identify those unique patterns in the DNA. So, let us say these are the nitrogen bases, and if you read from both the sides, you will see that it reads the same. So, this is an identifying mechanism of the bacteria to make cuts along the DNA or the RNA of the attacking virus. So, CRISPR-Cas9 is again it is just a name which signifies that we are using tools of nature to do gene editing. So, in CRISPR-Cas9 we are utilizing the restriction enzyme, and the name of that restriction enzyme is called as your Cas9. So, this Cas9 is basically the restriction enzyme or the molecular scissors. So, you would have heard that using CRISPR-Cas9 in 2018, a headline had come that a Chinese scientist He Junkai, he produced what is known as world's first gene edited babies. So, what he did in these gene edited babies, he took the embryos of those babies and he removed a particular gene in that babies. So, in human genome or the total set of genes, there is a particular gene which is responsible or which is supposed that it causes cancer, HIV, CCR5 gene. So, the presence of a particular gene, that is this CCR5 gene, leads to our vulnerability towards HIV. So, what this scientist has done, that he took the embryo after the in vitro fertilization, he took the embryo and he cut out using CRISPR-Cas9 this CCR5 gene. So, this CCR5 gene has been removed from those babies. Those were twins, they were known as Lulu and Nana. So when he removed this CCR5 gene, it is found that these babies cannot get HIV or they are immune to HIV in future. So again, so you would see that CRISPR-Cas9 always remains in news. So this year Alicia's case was in news, or Victoria Gray in US, she is having a particular kind of a sickle cell. And again, what we are trying to do using CRISPR-Cas9, we are trying to do genetic changes so that her body can start making healthy red blood cells. You would have heard in USA that a woman in US was cured of HIV using stem cell therapy. Again, in stem cell therapy, the stem cells that we extracted, we went for gene editing using this CRISPR-Cas9 only. So these gene editing tools that you keep hearing in the news, you need to understand the basics of that, then only you can answer many questions of this, questions in UPSC. And I have seen many questions in UPSC, they are of very basic understanding. They will ask you, can genes from one organism be shifted or put in body of another organism or not? So it is a very basic question that, see, we all living organisms, why is a bacteria or a virus able to infect us? Why is a bacteria or virus able to infect us? It is able to infect us because genetically we are very similar. In fact, all living organisms have evolved from a common ancestor. That is why all these organisms are very similar. That is, all organisms are using these ATCGs only to do the coding part. That is why we are very similar. That is why a bacteria is able to infect us. You would have heard about Bt cotton. What is Bt cotton? Bt is a term used for bacteria Bacillus thuringiensis. What is Bt? So this is a bacteria which is found in soil. So we have extracted something from a bacteria, we have put it in cotton plant, and now the cotton plant is itself producing a pesticide. So what we are seeing, that we can extract something from a bacteria and we can put it in plant. We can extract something from humans. In this case that you saw, we are extracting a human gene and we are putting it in a bacteria. So since all living organisms are very similar, or biologically and chemically they are same, they all are using this DNA as a method to do coding. It means that we can transfer genes from one animal or from one organism to another organism. That is the uniqueness of biotechnology. And this is what this is also known as the principle of biotechnology. So in USA, you would have also heard, this news had come, that a pig's heart was transplanted into a human. That is another case that he died after 2 months, but theoretically it is possible that we can extract something from pigs and we can extract or put it in humans. Similarly, we can extract something from humans and we can put it in other animals. So why? Essentially because we are all biologically and chemically similar. And what are the tools that we are using for doing these things? We are using these tools only. How do we put something in a cotton plant? How do we extract something from a bacteria? Again, in order to extract something from a bacteria to cut out the genes, we use the restriction enzyme. We will use this same method to transfer that desired gene into the plant. So this is a basic understanding of biology and biotechnology, and with this basic understanding, I assure you that if you look at last 7 to 8 years question, you will be able to answer all type of questions related to biotechnology. So with that, we will see more such topics in the upcoming videos and hope to see you soon again. So thank you all.